Hello and welcome to the third and final on-demand webinar in this regulatory series titled Global Development and Compliance, How to Stay Compliant and Collaborate Internationally, brought to you by Thermo Fisher Scientific and in partnership with the University of California, San Diego Extension Program. My name is Casey Pine, Senior Manager of Regulatory Affairs, Clinical and Compliance here at Thermo Fisher Scientific, and I'm happy to be joined by two experts today to help shed light and answer questions on international regulatory and compliance standards. First, I'd like to introduce Professor Ajit Sim. Ajit established Shiva Biotechnology in 1999 to provide regulatory compliance, quality strategy and support to the biotech, pharmaceutical and medical device industry. Ajit has worked with several multinational pharmaceutical companies over his 30 years in industry. He's a member of PDA and is an adjunct professor and instructor at SDSU and Reg Affairs. I'd also like to welcome Professor Naresh Nayar, an experienced regulatory affairs leader with broad experience in companion diagnostic, CMC and post-approval drug development activities. Currently, he is Vice President of Regulatory Affairs at Turning Point Therapeutics. Naresh is experienced in preparing teams for regulatory agency meetings and interacting with US and international regulatory agencies. With a combined total of 50+ years of experience as leaders in regulatory affairs in the biotech, pharmaceutical and medical device industry, both Ajit and Naresh are leading experts in the regulatory requirements for drug development and biologics. So welcome Ajit and Naresh. I'm happy to have you back for our last and final webinar in this series.
Thank you, Casey.
Thank you, Casey.
Naresh, can you provide a brief background on the growth in the companion diagnostic assays and the personalized medicine industry?
Sure. Thank you, Casey and Ajit. It's glad to be back. It's a two-part question, Casey. So let me give a little bit background on the CDX assay, and then I will talk about the global development of the CDX assay. The CDX assay, which is also known as a companion diagnostic assay, is defined as an in vitro diagnostic device, generally a class 3, which is used in combination with a drug or a biologics. You may ask, what is the connection between a CDX assay and a drug? I want to provide a very simple explanation. This is because some genetic or biological targets are not present in all patients with a particular disease. So the CDX assay can be used to identify patients most likely to respond to a drug or a biologics, as well as the patients who are at lower or higher risk for a particular side effect. This is interesting, as Ajit has pointed out in the previous webinars, that safety is of utmost importance to the health authorities globally. Let me elaborate on that a little bit for the safety for the CDX assay. I know everybody is familiar with the safety for the drug side, where you could have side effects. Even with CDX, you can have side effects. And let me elaborate what I mean by that. For the CDX assay, you have to take a tissue from the patient's where the infection or the cancer or any tissue that needs to be taken from the body. And that could pose a risk for the patient. So what happens is generally the regulatory agencies will ask the sponsor who's developing the companion diagnostic assay that whether this poses a significant risk or a non-significant risk. Now, if it is a significant risk, there's a risk to the patient for bleeding or for other side effects. And so they would require the sponsor to submit what we call is investigational device exemption, which is called IDE, abbreviated as IDE. It's almost equivalent as IND before starting a clinical study. And if the FDA deems that the companion diagnostic assay poses a non-significant risk, then you can proceed to the clinical study without the submission of the IDE. And generally, those devices are generally, as Ajit pointed out earlier, lower risk class 1 or class 2 devices. A little bit on the technical methods that are used for the CDX test, and it involves technologies like PCR, which is a polymerase chain reaction, or a reverse transcriptase PCR, protein expression, next gen sequencing, which is NGS, which we will talk about a little bit more detail later, uh, fluorescence in situ hybridization, which is abbreviated as FISH, and a mass spectroscopy and immunohistochemistry. More recently, there is a need for the use of a newer emerging technology, which is a liquid biopsy rather than doing a solid biopsy, which again goes to the fact that it is much easier to draw blood than to have a needle and poke the patient's organs or where the tissue, the cancerous tissue might be. I would say that the the liquid biopsy stage is where GS was a few years ago. So it's a very recent technology. So I will stop there for regarding the background, very high level background of the CDX assay. And if Ajit, you want to add anything more to that, I'll be happy to discuss that.
Yeah, thanks, Naresh. I think that was a good introduction to companion diagnostics. And as we know, from a regulatory compliance and quality perspective, uh, these companion diagnostics are very powerful tools that are used in what we call as precision medicine. And the reason they are very powerful is because they're able to detect certain, antigens, if you will, or what we call as markers. And one of the big challenges with medical devices, obviously, is that with companion diagnostics, um, specifically, they're also regulated not only as medical devices but as a pharmaceutical because they're used in concert with a pharmaceutical product to pinpoint a specific antigen of interest that the pharmaceutical can target. And just as a little background for those of you who are not familiar with medical devices, they're complex in the sense that they are classified by CDRH, one of the centers at the agency, called the Center for Devices and Radiological Health, and they're regulated specifically under 21 CFR 820, which is a regulation. And in the absence of a regulation, because these are relatively new tools, uh, the agency has, which is the FDA, has issued guidance documents, which is not necessarily the law, but it's a good idea to follow them because they are generated after a lot of discussion with industry experts and regulatory experts, and they generate these guidance documents, which is in the absence of the law. And it's a good idea to follow them because it's been vetted very thoroughly. So medical devices in general, Naresh, as we know, are classified into class 1, 2, and 3, with class 1 being just basic diagnostic tools, and 3 being the most sophisticated ones, which are used for sustaining life. And so these medical devices typically are class 2 unless you have software involved in them. And if there's software involved, then there are design controls required, so which makes them a little bit more sophisticated. So these tools have come across in a majority of cases in oncology, where it helps with precision medicine and identifying, as we know very well, that these medicines can be very specific to a certain population or demographic, if you will, and they are developed in a manner to meet those specific demographics or specific populations or disease conditions, if you will. So given that, medical devices still need to be regulated because I think it's very appropriate to say now with the pandemic, we have had a number of news instances with companies coming up with medical diagnostics for the detection of COVID-19, either the antigen itself or antibodies to it. And as we know, a lot of them have just disappeared, and the FDA issued a cease and desist, I think, order sometime back to about 19 different products that were supposed to detect, um, this antigen, COVID-19. And so why did they do that? Because developing a medical diagnostic is sophisticated because the percent success rate, as we call the false positive or the false negative rate, has to be minimal. Just imagine if an assay is not detecting it correctly. A simple example would be a pregnancy assay. A false positive or a false negative can be devastating in either case. So just like that, with a detection of a specific antigen, we want to make sure that the assay has been vetted out and developed properly. So the development starts very early on. So this gives us a lot of food for thought as to how robust these assays should be, because the false negative or positive rate cannot undermine the assay development. Naresh?
Yeah, no, thank you, Ajit. I think the last point is very key and is very important. As you mentioned, during this pandemic, there are many assays that have been uh, made available to people without the approval of the regulatory agencies. And I think, Ajit, you correctly pointed out that a lot of these assays are not specific enough to measure, and they are not specific enough in terms of the agreement. So what I mean by that is the FDA looks at or the other health agencies looked at the overall percent agreement when an assay is validated. So that has to be greater than 95 to 99% for the assay to be really available to the general public. So that's where a lot of the work that is going on between the health agencies currently and many companies who are developing the COVID assay, that to have the specificity of very high specificity with overall percent positive agreement as well as a negative agreement. They want the negative agreement to be as close to as the percent agreement. So thanks for bringing that, Ajit. That was a great point, and there's a great deal of discussion going on which assays should be used for general public especially for the COVID-19.
Correct, Naresh. And just to kind of elaborate a little bit, these assays, as we know, need two parameters. They need to meet two parameters: specificity and sensitivity. They need to be sensitive also, because the samples that we get, for example, they could be a blood sample, serum, plasma, or in the case of the pandemic with COVID-19, they can be buccal smears or they can be nasal swabs. Uh, and there are a lot of competing elements in that to make sure that the analyte that we are trying to detect is available and it is not camouflaged with something else. So it's not only sensitive, but we also need to make sure that it's available, to the assay conditions, right? Yeah.
Thank you, Ajit. Casey, let me take up the second part of your question, which was about the growth of the CDX assay. Uh, you know, I It is a relatively new phenomenon over last 10 to 15 years, and the most of the growth in the CDX, which is projected to be about market cap of about $10 billion by 2024. And if you look at it, about 15 years ago, it was less than $1 billion. It is driven primarily by personalized medicine, specifically in oncology, as Ajit has mentioned, and partly because a cancer is very personal. That means it is very specific to an individual. We could generalize it, but still it's very specific and it's very personal. And about 1 in 3 people in U.S. will be diagnosed with cancer in their lifetime. And in personalized and precision medicine, as Ajit pointed out, the pharmaceutical and the diagnostics are rapidly becoming integrated. What does that mean? That means, in practical terms, that the FDA or the global health authorities will approve the drug or the biologics along with the diagnostic, because they want to make sure that the benefit that the patients get by taking the drug is complemented by the companion diagnostic assay, which is tested for particular gene or a mutation. And very recently, there are many examples where health authorities have refused to approve a drug if there is no companion diagnostic, fearing that it might be used for, it might be a safety risk for the patients. And I think again, the pandemic is a great example of testing, of folks testing the COVID virus with an assay that is not either sensitive or specific or specific enough. And I do want to add that generally this statistics shows that you have a 30% chance of approval for a drug if it is submitted with a companion diagnostic, and about 8% without it. So right now, we talked about mostly in the area of oncology and maybe with the pandemic in the antiviral, but there are other therapeutic areas where the CDX are being developed in the CNS, in Alzheimer, in the many of the other blood disorders as well. So I know Ajit may have to want to add something to it, Ajit, regarding the specifically the growth of the CDX assay that you have seen, at least from a quality perspective.
Yeah, Naresh, in drug development, as we know, we're always coming up with ideas to make it more efficient, to ensure compliance from a safety standpoint, patient safety, as you also mentioned, as we have mentioned in prior webinars. Patient safety is of utmost importance, but more importantly is improving the quality of life. And as you mentioned, oncology is very personal. As we know that when biopsies are done, they find out they're either hormone dependent, hormone independent, drug independent, what works, what does not. In the early stages, I remember working on clonogenic assays, my gosh, back in the 80s. And clonogenic meaning that, for example, if you look at prostate cancer, which is highly heterogeneous, hormone dependent, hormone independent tumor, sometimes by administering certain cytotoxic drugs, they found that the tumor grows. Why? Because being so personalized, as we say, this topic of oncology, it impacts people differently. Is how do you improve the quality of life of the patient? Because certain drugs that we think may work actually enhance the tumor instead of shrinking it. And this is where this personalized medicine with IVDs or in vitro diagnostics comes into play. And and I think that there's a tremendous growth opportunity here for co-development, if you will, of drug diagnostic combinations. And as you mentioned, Naresh, very correctly, that there's a higher probability of getting an oncology drug approved if it is accompanied with a CDX assay, because then it becomes very personalized, if you will. And we started this, I remember, with personalized medicine right after 1979 with the introduction of hybridoma technology for monoclonal antibodies with Kohler and Milstein from Cambridge, UK, where this took a completely different turn on how we can get sensitive antibodies that are specific to the antigens to target them. Instead of polyclonal, you have monoclonal. So they're more specific and more sensitive. And so these assays need to be developed, if you will, very early on when you're developing the drug. So at companies, we need both of these elements, so you need the subject matter experts from both the drug development side and also from the diagnostic side that come together to be able to harmonize and make sure that we are able to come up with a co-marketed product that makes sense. Yeah.
Thank you, Ajit.
Thanks, both. You've given a lot of really good advice there. But I do want to ask about some of the, and from a Thermo Fisher point, we do have companion diagnostics in the oncology space around new technologies like next-gen sequencing. And while, the guidance is now available, it wasn't available at the time. So, these regulatory challenges, are still quite difficult, especially in emerging markets. What kind of advice can you give on some of these challenges when developing a companion diagnostic?
Thank you, Casey. Great question. So I think, the regulatory philosophy for developing the companion diagnostic, it is on a case-by-case basis because every diagnostic that is being developed is very different or is very specific to a particular disease. So I think in order to be successful globally, I think you have to plan early, talk to the health agencies in US, in Europe, in Asia Pac. For example, in countries like China, it is nearly impossible to get a human sample out of China. So you will have to think in a broader terms that either you partner with somebody in there or you have to develop the assay within that country, because China is a huge market for companion diagnostic as well as for different medicines. So that's just one example. It's just to be able to have a plan and talk to the health agencies early to what your target indication is and how you're going to develop the companion diagnostic. I think I'll talk about a little bit about later in the webinar about whether you want to develop a single assay, a single lab-based assay, or you want to develop a kit, because there is a difference between a single site PMA, which is called pre-market approval, which is just like the NDA, and a kit. So for a single site PMA, you have to send all the samples from everywhere in the world to a central lab, which can test it and then tell the doctors whether the biomarker is present or not. However, if you want to develop a kit which can be shipped to various places and they can use the kit as described on the package insert. So companion diagnostics, still there is no general guidance, there is no specific guidance. It is still on a case by case basis, and extensive collaboration and negotiations with the health agencies is required early in the development, because if you wait until phase 2 or phase 3, it is too late to really do a submission together with a new drug application and a pre-market approval application. So I think that was a great question. Thank you.
That's some really good advice there. Ajit, do you have any further comments around challenges beyond regulatory?
Yeah, there are manufacturing challenges, if you will. Manufacturing and quality go hand in hand, because any product that you manufacture must have and meet high quality standards, as I mentioned, sensitivity and specificity of the assay itself, or the test as we call it, Casey. But, you know, regulatory challenges are posed, and there are avenues to kind of mitigate those and overcome them. And that comes with experience and having the right subject matter experts like Naresh to talk to the regulatory bodies. Now let's look at some of the challenges related to development itself, if you will. So when we are working on a drug, in an oncology drug, for example, and we identify that to market this successfully, if we have a CDX in place, it would be very helpful. And as I mentioned before, we need subject matter experts between the two parties. So let's put that aside for a minute, but let's look at some of the challenges related to developing an assay itself, a diagnostic as we call it. One is we need to know all the different parts that go into the manufacture of the diagnostic. They're called components. Okay, and a component is defined by the US FDA as any material that is used in the manufacture of the diagnostic or the drug, but may not be found in it. But it is going to lend a tremendous amount of quality for the performance of that particular test, if you will, or the pharmaceutical product. So how do you control quality? And today, as we know, as we discussed in the previous webinar, we have global contract service providers, and they're all over the place today. And when we develop these assays early on, the R&D teams will identify a vendor, for example, who can make a specific component. And small companies, when they're developing a companion diagnostic, may not have the wherewithal to have a supply chain group, if you will. And the supply chain group comes into play because when you are identifying a component to be used in early stage clinical trials or even developing a diagnostic, only to find out that this diagnostic is going to be so successful that I need to scale up. And you find out that the small mom and pop shop that you have identified in R&D supplying a small component may no longer be able to supply you in large quantities. And they may not be interested in your quality group coming in to conduct a vendor audit. And that becomes a big challenge, and we have faced that before. So now you got to switch vendors. Switching vendors is a big challenge when you're in between in development. From, for example, if you're in phase 2 and you're trying to switch over, um, comparability becomes an issue. Okay, is it still sensitive? Okay, is my assay still sensitive? And is it still specific? So that becomes a challenge. So we need to identify our manufacturers who are part of the supply chain, if you will, very early on to make sure that they are robust in their capabilities of providing high quality components to us. Okay.
Thanks. You've both given really good advice just now on development and strategy. What about advice to create buy-in from a clinical team for a diagnostic strategy, particularly one that might be initially resistant?
Hi, Casey. That's a great development question. If I can just go back quickly to Ajit's comments, I want to summarize it in a few words or lines or a paragraph. In my opinion, at least from a regulator's perspective, and Ajit has mentioned this, but I want to distill it to a few things. A diagnostic product is a complex system of reagents, instruments, software, algorithms, and procedure, all of which comprise the final regulated product, as Ajit mentioned. And all the components and steps are monitored and evaluated by the regulatory agencies and globally. And we have highlighted some of the challenges, developmental, regulatory, and commercial. So I think that was, I just wanted to bring that up. Now, coming back to your question, beginning the discussions with the clinical development team early is a key, because you want to align on the intended use. Early in the development, it's not clear whether the research assay that you have in the lab is going to be appropriate in a clinical setting. So that's number one. And then you have to decide whether you want to use the assay working with the clinical team, whether it is selective, whether it is predictive, or it's prognostic. Only in the case of selective, where you will select the patients for a study, you will want to develop the companion diagnostic assay. So I think you want to align that with the not only with the clinical team, but with also with the development and the commercial teams. Uh, you have to work with the clinical team or development team that whether we're going to submit the new drug application with the PMA at the same time, or your strategy is to your clinical data for a particular indication is strong enough that you can negotiate with FDA that we will get the drug approved first, and then as a post-marketing commitment, you will develop the assay or submit the PMDA maybe 6 months later. So I think that is a case in most of the companion diagnostics, because a lot of these drugs coming out of oncology or other therapeutic areas which are personalized have a very strong response rate. They have provided a great deal of benefit to the patients, and FDA and the global health authorities are flexible, say, okay, we will approve your drug, but you must provide the pre-market approval application for the companion diagnostic after 6 months. And then you have some development challenges with the clinical team, depending on the strategy that you will use, how the samples will be collected, whether they will be shipped to a testing center, whether, as I mentioned, is it going to be a centralized lab, or it will be multiple labs who will be using kits. And in my experience, Casey and Ajit, I think it is best to leverage the business development group early on, because a lot of the small companies, they don't have the bandwidth or the resources and the money to develop, or the expertise to develop the assay in-house. So you have to go outside the company to really be able to transition from a translation early development, what I call is a lab developed test, to transition to a CDX. So that you need to leverage the business development group. You need to leverage the translation group to really understand whether the assay is transferable to an outside vendor. And finally, you need to talk to the clinical and the medical folks. What I mean by that is key opinion leaders. So it is very important to talk to the key opinion leaders, that whether they will be able to, what is their feedback on this assay we are developing for a particular indication, that can be incorporated into the protocol along with the clinical team. As we have mentioned many times on the webinar series, it's all about the patient safety and providing maximum clinical benefit to the patient, especially if it is directed towards a particular biomarker where the response rates are higher. And summary, my experience is the companion diagnostic strategy or development should be a part of the broader clinical development strategy, and it should be shared across the company so that everybody is aligned with the strategy. Senior leadership team and Ajit, or experience on your teams who have experience of developing the companion diagnostic assay, working with the global health authorities, not only in the US, in Europe, and also emerging Asia Pac countries. Thank you.
Ajit, do you have anything more to add to that question?
I think to kind of conclude this, I would just say that drug development and medical device development is a multifaceted approach. So we have many moving parts, different departments, right? We have the research and development for both components, for the pharmaceutical products, as well as the medical device or the companion diagnostic. They're completely different groups, right? Because the R&D is very different. And then you have a regulatory group that should have experience in both those areas. Then a quality group that is able to drive compliance, across both components. And then analytical groups, which are different for the medical diagnostic and the pharmaceutical product. And then you also have the manufacturing groups that eventually take over the process from both R&D groups and either contract them to the medical device manufacturer or the pharmaceutical product manufacturer. And they're typically two different companies that do this because they have expertise in those areas. And then there's a single supply chain group that will ensure continuous supply of qualified vendors who are either under contract with a quality agreement or articles of commerce, which we buy from companies that need to be able to provide the component, meeting those predetermined requisites, if you will. Then you have the clinical group, which plays a very pivotal role because they have a clinical program where they have the clinical protocol for both the um, medical device as well as the pharmaceutical product. And they both need to come together because the patient comes in and they have to meet the IE criteria, the inclusion exclusion criteria, if you will, and they're screened, they get enrolled. And of course, after they go through all the informed consent and the ethics review and all that, the IRB approval and all that, we need to make sure that the samples are stable. They're able to go to the central lab, as Naresh mentioned, so where they can be analyzed, with the development, with the CDX that we are developing as a companion diagnostic, and they're effective. Because at the end of the day, to get the drug along with the companion diagnostic approved, a lot of data is generated, and all of this has to be mined very carefully. So we are looking at, a couple of things here. One is the p-value. Okay, is it safe? Is it efficacious? Right? And two is, did it meet the endpoint? Because that's the most important thing. So I think all of this put together makes it challenging, but makes it also very exciting that we're able to get a cutting edge therapy out to the patient population.
Wonderful. Thank you both. I do want to ask another question before we wrap up today, and That's what's the state of our current companion diagnostic technology.
Thank you, Casey. A great question. I think I mentioned in my previous response to the first one regarding the background on the assay, some technologies like FISH, IHC, or PCR. Now, these technologies, Casey, they can only analyze one target gene at a time. Uh, and the latest technology that's being used extensively and being promoted by many companies and health agencies is the next gen sequencing. It is also called NGS. NGS can process hundreds to thousands of genes and detect multiple biomarkers simultaneously. So you imagine the efficiency with that. So I think that is the latest technology that is being heavily used to really promote the development of the companion diagnostic assay and bring value and safety or a benefit to the patients. The conventional wisdom is that if you use a standard tissue biopsy, sometimes it's feasible, but it's risky depending on the tumor location. So very recently, many companies are also developing a liquid biopsy that is using a blood sample rather than a solid tissue. A couple of advantages with that is that it's non-invasive. It still uses NGS technology, but also to really test the solid tissues with NGS, it requires tissue preparation, paraffining them, and then sending them to the location and testing. So generally, the turnaround time is about depends on the company's 10 to 14 days. And whereas the liquid biopsy using NGS, you can draw the blood and you can get the results within 2 days. So you can imagine a patient who is really waiting to know the results of the biomarker. Liquid biopsy really is very attractive. But again, the liquid biopsy, as Ajit mentioned before, sensitivity and specificity is not at that level whereas it's for the solid tissue. But we hope that in the future the liquid biopsy will be widely used, but at this point both of them have to be used simultaneously to first analyze the biomarker and then also to provide development to the liquid biopsy, because you want to see the concordance, right? You want to get the same result from the NGS using a solid tissue versus a liquid biopsy. So I'll stop right there, and without going into too much technical details of the liquid biopsy, but those are the two really upcoming technologies. I mean, technology is NGS, but the technique is the solid tumor or versus the liquid biopsy. I think it's a fascinating area to be in right now. And very recently, FDA approved the first test using the liquid biopsy. So it's making progress or gaining acceptance amongst the regulatory agencies. Thank you.
Thank you. It's definitely an exciting time. So thank you both for your time and insight today. We've covered a lot of great information, and we also appreciate everybody joining in. If you enjoyed today's discussions and are looking for some more regulatory insights, please register for our on-demand webinars in this series. Lastly, thank you to both of our partners at the University of California, San Diego Extension Program, where both Naresh and Ajit are professors of regulatory and compliance. Thank you.
Thank you, Casey.
Thank you, Casey. Thank you, Naresh.